Dr. Muthamala sir.
Am I audible?
Doctor, can you hear me?
Yeah, I can hear you. I mean, am I audible?
Yes. We'll begin. Okay. Department of Plant Science, School of Life Science, University of Hyderabad. Cambridge University, right? New Delhi. His major area, research area are plant molecular Nasi egg scientist practice department. Inspire Faculty Award from DST, Ministry of Science and Technology. Fulbright Nehru State. Sorry, there was some technical issues. Hello. Am I audible?
Yeah, you are audible now.
With this short introduction, I will like to invite Dr. Muthamala to deliver his lecture. The floor is yours.
Thank you. So I'll just share my screen. There is some issue with my video camera, so it's not able to share my videos.
Yeah, okay. It's okay.
Thank you, sir, for giving me this opportunity to talk to the students about CRISPR genome editing. Just an overview of what is CRISPR, and since I am from the Department of Plant Sciences, I will be talking more about plants. I mean, not more, it is all about plants. So where CRISPR has been successfully used in improving few important traits. So basically, I'll start with why we need such technology when nature has already given us good things. And the second thing is that what CRISPR is and how it is being used and what is its acceptability in India. A little bit of policy issues also we will discuss in the next 1 hour or so. To begin with, just to lay a foundation to the audience about why we need such technology. If you know, today morning around 9 o'clock, I have taken the screenshot. It's showing that the current world population is 7.9 billion. The current world population is 7.9 billion, which is expected to reach 10 billion by the year 2050. So by 2050, we are going to be 10 billion, right? And since our kindergarten, we would be learning about the basic needs for a human being. We all know what are the 3 basic needs of a human, right? Food, shelter, and cloth. Now, if you ask the question whether all the 7.9 billion has got all the 3 basic necessities, whether all the 7.9 has got all the 3 basic needs, the answer is no. Food is still a distant dream for several of us. If you see the statistics, if you see the World Hunger Report, I'm going to show you a little more details about this World Hunger Report. It says that 1 in 10 among us goes to bed with an empty stomach. So that accounts for 821 million. And in case of children, for every 10 seconds, a child dies from hunger-related causes. And 1 in 4 of world children suffer from malnutrition. This is the scenario. I don't know how many of you would be remembering this picture. This picture was clicked by a famous photographer called Kevin Carter in the year 1993, which shows that a hunger-stricken child is trying to reach to the United Nations feeding center to get some food, whereas at the background, a vulture is waiting for the child to die so that it can feed the dead body of the child. This is a scenario of South Sudan in the year 1993. It is going to be 2023 next year. If you ask whether the situation has improved, the answer is no. If you see the global hunger index by severity, several countries in Africa are still suffering from hunger related issues. Even India is not green. India is still in a range what they call as serious. In India, hunger is still a serious issue which has not been addressed so far. Are we making good progress? Say, for example, India is lying in South Asia. In 2000, our hunger index was 38.1, and in 2021, last year, it is 26.1. The progress is also made. The progress made is also very slow in this regard. So, whenever you get time, I would recommend you to read this Global Hunger Index 2021 report, which has very interesting facts about how hunger is prevalent among the global population. This is the case of adults. When you come to the case of children face 3 issues. Children means those who are under the age of 5. Our children faces either stunting means too short for their age, wasting means too thin for their height, and overweight means obesity. Particularly the stunting and wasting statistics, I think I'll show you a little bit of statistics about the stunting. Stunting, if you see, India is pink in color. Pink means in India, stunting is very high. Our children under the age 5 are stunted, where in India and few countries in Africa. Right, that is where the United Nations has come up with the 17 sustainable development goals. I hope many of you would have been aware of these 17 sustainable development goals. One thing, if you notice very closely, that out of 17, 3 are directly related to food. No poverty, you have to have food. Zero hunger, you have to have food. Good health and well-being is directly related to food. So 3 out of 17 SDGs are directly related to food. Why? The answer is because the world checklist shows that there are 11,28,000 vascular plants, out of which archaeological evidence says that our early human beings were consuming approximately 7,000 crops. Our early humans, throughout the history, they were consuming approximately 7,000 crops. This number slowly got reduced, and today we are ended up with just 3 crops: rice, wheat, and maize. If you ask a question to you, what you had in your breakfast, the answer would be either rice or wheat. What will you have in your lunch? Either rice or wheat. What will you have in your dinner? Either rice or wheat. So slowly the 7,000 got reduced into just 3 crops: rice, wheat, and maize. Although we are not using maize prominently in India, rice and wheat constitute a major proportion in our plate, our diet. These 3 crops, along with other 12 species, leafy vegetables and other vegetables, tubers, etc., plus 5 animal species, cater to 75% of the world's food. So what we are consuming as such from plants, only 15 species are being consumed by the humans. So now you can ask a question that on one hand we are having 7.9 billion people, on the other hand we have just 15 crops. So with the 15 crops, how will you cater to the nutritional requirements of the 7.9 billion population? The answer is it's not possible. Right? It's not possible, that too in normal situation. But now we have global climate change, agricultural lands are becoming infertile, rapid urbanization is happening, many agricultural lands are already turned into cities where homes and apartments are being built, and the waters are getting dried up, our rivers are not running with sufficient water. So groundwater level has already decreased, with so many issues in agriculture. Feeding 7.9 billion population with the current agricultural practices is nearly impossible, and that is where hunger is prevalent. That is where malnutrition is prevalent. That is where we still have 2 issues in the world. One is hunger, the other one is hidden hunger. What is hunger? Hunger is what you feel when you don't get sufficient sufficient food. What is hidden hunger? Food, you will get food, but that food will not be able to cater to the nutrition requirements of your body. So it is not just the problem of getting food, the issue is getting nutritional food. And that is where we say that biotechnology can improve the existing crop species, where today I'm here to talk to you about genome editing. Right? We are saying that if we edit few genes or few portions of the genome, we can get improved varieties. Is it true? The answer is yes. I'm going to show you a few examples or case studies where they have shown that editing few parts of the genome helps in improving the crop. It helps in improving the yield, it helps improving the resistance, it helps in several ways. I'll show you a few case studies in agriculture, but before getting into that, to know what to edit. Say for example, if I'm going to edit this nucleotide, I should know what is this nucleotide, where it is present, in which chromosome it is present, what is its exact position, and those details we have to get. So editing the genome is relatively easy, but finding the target is tough. Right? A genome will be having thousands and thousands of genes. Of those genes, which gene you are going to edit is the question. To find that answer, to find the answer to that question, you have to have the complete detail of the genome, and you should have characterized the gene properly. So that is where functional genomics plays a major role, although I'm not going into the details of how it is done. One good way, or the straightforward way, is genome sequencing. I don't know how many of you are aware of this next generation sequencing. So now what we can do, we can isolate the DNA and give it to the company. Company will sequence the DNA and they will give you the sequence file, right? So all what we can do, we can isolate the DNA and give the DNA to the company, and these companies do sequencing and they use different platforms. These are the 2 prominently used platforms today. Although, I mean, people also use PacBio platform. I'm not showing it here. You know, the sequencing has become an advanced technology where the DNA is read to identify the nucleotides present in the DNA. So if we are giving a genome DNA, they will identify the nucleotides present in those, nucleotide sequence present in those in that DNA sample, and you will get a file like this. Then we have to do intense bioinformatics analysis to make sense out of this. DNA is nothing but 4 letters, right? A, T, G, C. So in the file, what the company gives will be having these 4 letters only. But how will you identify which is a gene, which is a UTR, which is a non-coding element, which is a transposable element, etc., etc. So there are several bioinformatic pipelines available to make sense out of the sequencing file. So I'm not going into the details, but all we can do is we can identify the genes as well as the non-coding regions using different bioinformatics pipelines. So once we have identified, we can easily identify the candidate gene that can be targeted for editing. We can also edit the upstream regions, what we call as promoter elements also. So promoter editing is also prominent, as well as gene editing is also prominent in the present day science. So once we have identified which gene we are going to edit, then we are going to use the CRISPR-Cas9 technology, right? So now, once we know the target, editing the gene is very easy. How are we going to edit the gene? We are going to use Cas9. So Cas9 is nothing but a protein complex which has this protein. The protein has 2 nucleus domains and it has a guide RNA. Right, so Cas9 is a protein that has 2 nucleus domains and it has a guide RNA present within it intact. Now, what we can do, this guide RNA sequence, if the sequence is complementary to the target DNA sequence, if the guide RNA sequence matches with the target gene in the genomic DNA, what happens is this Cas9 protein along with the guide RNA binds to the DNA and it glides through it till the place where it achieves a perfect complementarity. Once a perfect complementarity is achieved, what will happen? As I said, this Cas9 protein has 2 nucleus domains. These 2 nucleus domains introduce nicks in both the strands. So now the DNA is broken. Right. This broken DNA undergoes repair. Mostly this non-homologous end joining repair happens where some random bases are inserted into the DNA. So when random bases are inserted into the DNA, the gene gets deactivated. Right, this is how Cas9 protein works. So this is what we call as CRISPR-Cas9 based editing of the target sequence. Let it be a genome sequence, gene sequence, or upstream element. Right, it can be of any part of the genome can be sequenced. Only thing is that the 3 bases are important. This is what we call as PAM sequence. PAM sequence. So we have to identify the PAM followed by the target sequence. So we don't have to go into the details of the CRISPR-Cas9. As of now, all you should know is the basic mechanism by which the Cas9 works to introduce nick in the double-stranded DNA, and the double-stranded DNA, which is now broken, undergoes repair where random bases are inserted. Right, this causes structural change in the genome, and this might have an effect in the gene's expression. Right, so I'm now going to show you a few case studies where people have shown that editing few genes result in very good benefits. So the first example, first few examples will be on editing for increasing the yield. Now we know that we have to increase the yield of plants because we have to feed more number of people. Right, we have 7.9 billion people waiting for food, and agriculture is the only source of food grains. So increasing the yield is very much important. Increasing the yield of cultivated crop species is very much important. So to do that, there are several approaches being used. Now that CRISPR-Cas9 technology is there, people even tried using CRISPR-Cas9 to see if CRISPR can do any good. One study by Wang et al. In the year 2019, I'll show you, there are 3 genes in rice. OS stands for Oryza sativa, which is the scientific name of rice. GS means grain size. GW means grain width, grain weight. And GN means grain number. So what they are doing, they chose 3 genes, GS3, GW2, and GN1A, and they edited those 3 genes using CRISPR-Cas9. So they used a vector. I'm not going to the details of the vector. What they did, they edited these 3 genes, GS3, GN1A, and GW2. Right. When they edited, if you see, when all the 3 genes are edited, this is a triple knockout. When all the 3 genes are edited, compared to the wild type, the grain size and grain number has increased. The grain size and grain number has increased. So this is the difference between the wild type as well as the edited line. They edited 2 different cultivars. One is J809, the other one is L237. So in both the cultivars, they achieved better grain yield. Right. So here you can easily see the difference. Yeah. So this is the wild type, and this is the triple knockout. So wild type, the panicle size is small, whereas in the knockout, the panicle size is large, as well as it had more grain number, grain size. So when they arranged 10 grains, you can see a clear difference. The grain size has increased, panicle height has increased, and this has resulted in the increased yield. So just by editing 3 genes in rice, people are able to achieve higher grain yield. Another example, in the previous example, 3 genes were edited. Here only 1 gene is edited. It is an amino acid transporter protein, OSAAP3. This is also in rice. So what they did, they edited this gene in 2 cultivars. One cultivar is ZH11, the other one is KY131. I'm not going to the details of these graphs, but if you see, compared to wild type, the edited line has more grains in both the cultivars. In ZH11 also, in KY131 also. So grain yield per plant increased tremendously when a single gene called OSAAP3 was edited. I'm not going to the mechanism. Yeah, this is in wheat. Rice is a diploid crop, so editing a diploid crop is very easy because only 1 copy of the genome is present. But wheat is a hexaploid and it has 3 genomes present in it. I don't know how many of you are aware of, but if you don't, please note that wheat has 3 genomes: A genome, B genome, and D genome. That is where wheat is a hexaploid. So if we have to edit any gene, we have to edit that gene in all the 3 genomes to achieve 100% editing. So what they did, they targeted this gene CKX2 and they achieved editing in all the 3 genomes. The result is very good, if you could see. There is an increased yield in the Cas9 edited line compared to the wild type. Right, a single gene editing in wheat resulted in increased yield. So these are the three case studies which have shown that increased yield could be achieved through CRISPR-Cas9. Now let's see a few examples where quality is being improved, improving the quality. One issue with wheat is pre-harvest sprouting. What the farmer does, once the farmer harvests the panicle, and when they store it, what will happen is if the temperature and the humidity is ambient, the wheat will start germinating. If it germinates at this stage, it's of no use. The farmer will run into a loss. Right? This is what we call as pre-harvest sprouting. So before harvesting, they have removed the panicle. They have to harvest the wheat grains out of this. But before they do that, sprouting starts. So this always results in a huge loss for the farmer. So how to arrest this sprouting is the question, where people identified one gene called QTL for seed dormancy. The gene's name is QSD. QSD stands for QTL for seed dormancy, again in wheat, Triticum aestivum. So one gene they edited in all the three genomes, A genome, B genome, and D genome. This is the confirmation of edited lines. I am not going into the details of it. I want to show you the result, how good the result is. When all the three genes are edited, they saw that there is 100% arrest of sprouting. You can easily compare with the wild type. Wild type sprouting is there, whereas in the edited line, just a single gene editing, there is no sprouting. Right? Although the plants behave the same way, the plants look the same, the plants height, yield, everything look the same. But only the best part is when this gene is edited, there is no pre-harvest sprouting. Right? What they did, what actually happened in the gene, the seventh nucleotide, T, C, 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 T, G, there is an insertion here. So the CRISPR-Cas9 inserted one nucleotide in all the three genomes. That resulted in knocking out of this gene QSD1. That resulted in the arrest of pre-harvest sprouting. Right. Similarly, in maize, Zea maize, they edited a gene called WAXI. WX means WAXI. When they edited this gene, they found that the gene's quality improved in terms of its amylopectin content. So starch is very much important, particularly maize. Industrial starch extraction from maize is a very prominent one, right? So starch content should be more in maize for industrial usage purposes. So to improve starch, what they did, they edited a gene called WX. WX stands for WAXI. WAXI. When they edited, they found that the seed contained more starch. In rice, they edited two genes, SBE1 and SBE2B. SBE stands for starch branching enzyme. So when they edited these two genes, SBE1 and SBE2, what they found is when this SBE2 was edited, the grain was totally opaque. When SBE2 was edited, the grain was totally opaque, what is the reason for this phenotype? The reason is it has become rich in a particular kind of starch. The starch is called resistant starch. To give you a basic information about the starch, There are three types of starches present in every grain. One is rapidly digestible starch, second is slowly digestible starch, and third one is resistant starch. Rapidly digestible starch gets digested very fastly, resulting in the release of glucose in the bloodstream immediately after consumption. Rice is a very good example of high rapidly digestible starch content grain. Slowly digestible starch, where the digestion takes very slow, little slowly. Okay, the last starch is called RS. RS means resistant starch. As the name indicates, this resistant starch resists digestion in the small intestine. So in the small intestine, it won't get digested. It will be moved to the large intestine. In the large intestine, the gut microbiota catabolizes or breaks down the resistant starch into short chain fatty acids. And those short chain fatty acids will be released into the bloodstream in a slow and sustained way. That is where for diabetic patients, people suggest oats, because oats is rich in resistant starch. So immediately after consumption of resistant starch, your blood glucose level will not get shot up. Whereas in the large intestine, upon the action of gut microbiota, there will be a slow and sustained release of glucose in the blood. So here, by editing a single gene, starch branching enzyme 2B, they achieved high resistant starch content in rice. If you could see, RS means resistant starch. You could see the wild type has very less resistant starch, whereas the edited line has high amount of resistant starch. So this rice would be very good for diabetic patients. Now coming to disease resistance, people also showed that editing a few genes can confer resistance to diseases. I'll show you a couple of examples. Three genes in rice, three genes belonging to a gene family called sweet proteins. Okay, these proteins are called OS sweet. OS, as I said, it is Oryza sativa. So sweet, three genes, gene 11, 12, and 13. So these three genes are edited in rice. So what they did, they generated the guide RNA that targets these three genes, and these genes were edited. Now I will show you what happened in edited line. These are the wild type. So this is the wild type. Wild type is susceptible to blast disease, BLAST, caused by Magnaportha oryzae. Right, when they edited, if you could see, these three are the edited lines. In edited lines, the infection is confined to the leaf tip only. The infection could not progress into the leaf. So just by editing three genes, what they achieved? They achieved a way to stop the infection only at the tip. It could not intervene into the rest of the leaf, so that the rest of the leaves will be healthy, it will be performing the photosynthesis or whatever the processes that they are supposed to do. So the plants will be normal. Right. Similarly, in wheat, they edited a gene called EDR1 for another fungal disease. I forgot the name of the disease. But when they edited, they found that the fungal infection is limited. It is not totally limited, but if you could see, this is the wild type. This is the edited line. But compared to the wild type, in edited line, the infection is less, although it is not 0% less. To an extent, disease containment was possible. Similarly, in another study, an elongation factor EF4G was edited in rice, which resulted in resistance to virus. Although in this paper they didn't provide any pictures, so I'm not able to show you how virus infection is restricted, but yeah, the paper says that when the gene is edited, this EIF4G is edited, the edited lines shows resistance. Similarly, herbicide tolerance is also being shown using CRISPR-Cas9. One gene is ALS1 in rice. When they edited this gene, the plants looked similar as compared to the wild type, but these plants showed resistance. The edited lines showed resistance to herbicides. A tubulin A2 gene was edited in rice. Again, the edited lines showed resistance to two different potential herbicides. One is pendimethalin. So if you could see, wild type plants were not even able to germinate in the presence of pendimethalin or trifluralin. But the edited lines were growing normally. So herbicide resistance could also be achieved in rice if we edit tubulin A2 gene. Another interesting feature or another interesting application of CRISPR-Cas9 is domestication. I don't know how many of you are aware of this terminology, domestication. Whenever we study the origin of species, we come across three terms: evolution, speciation, and domestication. Plants evolved, humans evolved, other organisms evolved, all the biotic components of this earth evolved. They are still evolving due to different pressures, what we call as evolutionary pressures. There could be different selections, positive, negative, or neutral, which resulted in the formation of different species. Then what humans started doing is humans started domesticating. Humans tried domesticating and they were successful in it. Say, for example, the present day dogs. Dogs are not the product of nature. Dogs are domesticated from wild animals like wolves and foxes. Right. Similarly, crops were also domesticated. Domestication took years, hundreds of years to get what they wanted. If you see the wild rice, there is no good grain in the wild rice, but the domesticated rice, the grain size and the grain shape is good. If you see the wild carrot, you won't even believe that whether people would eat this carrot or not. It looks like a root. But if you see the present day carrot looks so delicious. If you see the wild tomato's shape of the fruit were of the size of peanut. But today the tomato size is huge. If you see the size of the tuber, the wild potato, it is too small. But the present day potato is too big. Right? So people did domestication to achieve better traits. But this domestication took years and years. And this was purely based on selection. They selected for the trait what they wanted. So what has happened is, say for example, the wild type, the wild has tens of thousands of genes. Of these, say for example, 20% of the genes are beneficial for stress resistance. In the process of domestication, the domesticate would have got, say for example, some 10,000 genes. several informative genes or important genes would have lost during the process of domestication. That is where wild will be resistant to diseases, wild will be resistant to other environmental effects, whereas the domesticated crops are susceptible to diseases. Domesticated crops cannot withstand environmental effects. So what people thought is, let's redo the domestication. That is what we call as de novo domestication. So what they did, they demonstrated in tomato. They took a wild tomato called Solanum pimpinellifolium, which has indeterminate growth. The fruits are pea-sized. Okay, the fruits are too small. However, it is stress resistant. So what they did using CRISPR-Cas9, they edited 6 genes. 1, 2, 3, 4, 5, 6. Just 6 genes, which resulted in the formation of fruit, which looks like the present day tomato, Solanum pimpinellifolium domesticate, which has a determinate growth, large and more fruits, 50% high lycopene, and they are stress resistant. So in this de novo domestication, no gene is lost, except that a few genes are mutated. Similarly, people tried doing de novo domestication in rice, although this paper is not completely available. So I'm not going into the details of this. Yeah, only thing is that what you have to understand is you assume these colored blocks as genes. So wild species has rich, or wild species are rich in genes. So what happened is, during domestication, we lost several informative genes. The modern varieties have very less genes. That is why wild are able to withstand any stress, whereas the modern varieties are not able to withstand any stress. Right? One study is showing that to produce 1 kilogram of rice, we need approximately 3,000 liters of water, whereas the wild doesn't require that much amount of water. Why? The wild has a good number of genes that enables their growth, development, and reproduction even in less water, whereas the present day crop requires more water because it has lost those genes. So how you will bring back those genes? You can use de novo domestication using CRISPR-Cas9. Right. So I'm not going to the details there. The problem is whether public are going to accept or not. When GMO was introduced, when BT cotton was coming into India, there was a huge opposition. Even now, you would have seen in the last 2 years or 3 years ago, there is a huge opposition for BT brinjal. So whenever the technology tries to deliver a better product, there is an opposition. So GMOs are almost banned in India, except only one GMO that is allowed till date, that is this BT cotton. So what will happen if we develop better varieties through genome editing technology like CRISPR-Cas9, but if the public acceptability is 0, then it is of no use, right? Few countries have already started accepting genome edited lines. India, although the slide is an old slide where it is showing discussion is going on, luckily in March 2022, we exempted genome edited plants and we now approved for commercialization of genome edited plants. Right. So now India says that genome edited plants will not be considered as GMOs. It can be commercialized. The genome edited lines can be given to the farmers. That is where the Department of Biotechnology under the Ministry of Science and Technology released last month they released a guideline for the safety assessment of gene-edited plants. So before releasing anything to the farmers, we have to follow all these guidelines. And once we ensure that our seeds or our procedure complies with the guidelines, then it can be released to the farmer. Right. Why we need such a technology? Because our national goal is we need a low input but high output agriculture. What is low input? We should spend little money in agriculture, but we should reap maximum harvest out of agriculture. We should give less fertilizer. We should give less water. Whatever the input that is going into the field should be less. But whatever the harvest that we are getting out of the field should be more. That is what Professor Deepak Pental of Delhi University, he says this, that our agriculture should be low input but high output agriculture. But in Indian scenario, it is the other way around. A farmer has to spend more here, but whether he is getting a good output or not is a question mark. That is where many farmers are giving up on agriculture. Many farmers are not even encouraging their next generation to take up agriculture as a profession. Many farmers, they are selling away their agricultural lands because there are several issues associated with agriculture. The most prominent one is climate change. India is getting dried from the middle and it is extending towards the coasts or towards the border. And remember, India has 2% of world's land, 4% of fresh water, but its population is high. Right? Plants cannot grow in sewage water or sea water. Agricultural plants need fresh water as much as a human being wants it for his survival. So there is already a tug of war between the agricultural plants as well as the humans. If you take any conflict that arises between two states of India for sharing the river, you will get to see the root causes. One state will be needing it for agriculture, whereas the other state will be needing the fresh water for their population. Right. So climate change is posing us a heavy challenge to this freshwater resources. When there is no water, drought is going to happen. If you see the damage caused by drought is too high, whereas the other damages, even if you sum the rest of the damages, it won't reach this much. Right, so it causes a huge damage. When the global temperature goes up, the waters are going to get dried. The water table is going to go less, and it is going to cause a serious issue. That is why we are now using different technologies, including CRISPR-Cas9, to develop crops that can grow and reproduce in less amount of water. We are already seeing that farmers are suffering because there is no rain. There is a late monsoon or heavy monsoon. There is an increased impact of pests and insects. Diseases are there and other natural calamities are there. That is where the 2019 data of this National Crime Record Bureau, it says that in a year, these many farmers and daily wagers committed suicide. So if you divide this number by 365, you will get to know what is the death rate per day. Right. So that is where they say when all the farmers are gone, it would be anarchy. Right. So farming has become a killing field because there is a loss. So this is going to continue because FAO is predicting that by the year 2050, crop yields are going to decrease by 25% if we don't address climate change now. And remember, addressing climate change is not easy, but using technology to improve the crop yield is easy. is relatively easy. That is where we have different technologies, including genome editing technology, in our hand, which can deliver good or improved seeds to the farmers. And that is where we have to harness such technologies for the betterment of agriculture and to secure the life and livelihood of the farmers. And with this, I'll stop my talk here. If you have any questions, I'll be happy to address. Any questions?
Thank you, Dr. Muttam. The session is open for discussion. If you have any questions, you can ask. So there is a question in the chat box.
Okay. Can the Cas enzyme translate easily when applied to brain and medical entities? Actually, I'm not sure of this thing because we work on plants, so we are more into plant system. So somebody from animal sciences will be able to answer this question.
Any other questions? There is one more question in the chat box.
So far, there is no study that shows that there is an effect. And in my opinion, also, it's not going to affect the gut microbiota. Unless the composition of the seed changes, it's not going to affect the gut microbiota. It won't affect.
Next question. No more questions, then I would like to thank Dr. Muttam for joining us today and elaborating about the CRISPR genome editing techniques and giving us various insights about the crop improvement and the need for the crop improvement in today's scenario and in the future. Thank you, Dr. Muttam for joining us.
Thank you, sir. Thank you.